Hello everyone and welcome to my channel Be Yourself. This is Dr. Rajni and today I have started a new module, that is the biochemistry, and this I will first deal with the biomolecular interaction, because whenever we call about the biochemistry, it always means that how biomolecules, or basically we can say that what is the chemistry behind the biological molecules or the biomolecules. So let us start it without any delay. So in this module we will be dealing about the covalent and non-covalent interactions, how they are interfering in the interaction of different biomolecules. Next, we know that hydrophobic and hydrophilic interactions are very important in case of biological world, and this is basically with respect to their solubility and their interaction with different polar components which are present inside our body. So next topic will be our hydrophobic and hydrophilic interaction, and how all these bondings are influencing the biomolecular structures of the living world. So let us start with the first one, which is the covalent and non-covalent interactions. So before dealing with this, we must know where do these biomolecules lie and what are the evolution of these biomolecules. So you all know that atom is the smallest particle which can exist, though there are the nucleus and electrons are also there, but they cannot exist independently. So we consider atom as the smallest particle which can exist independently, and an atom consists of a nucleus around which electron revolves around. Electron is the revolving present on the orbital of the nucleus of the atom, which is constituent of the nucleus, and the nucleus consists of the proton and neutrons. So when we will look into it, so though you already were knowing all these things, but just I am giving a quick recall of all these things. You all know that mass of the neutrons is 1.75 into 10 to the power minus 24 gram, whereas that of the proton is 1.7 into 10 to the power minus 24 gram. So all these things are well known to you, but as compared to the neutrons and protons, the mass of electron is almost negligible. So we consider proton and neutron as the mass number of any atom, and we know that an atom to be a neutral in conditions, it should contain an equal number of proton and equal number of electrons. And because of this reason, number of protons is sometimes not sometimes basically considered as the atomic number of the atoms, and mass number can also be expressed as the atomic number plus the number of neutrons present in the atom. And a number of atoms is equal to the number of protons, as we can see here, and as number of proton will be equal to that of the electron until unless there is not less loss or gain of electrons, and in that case number of proton will be equal to that of the number of electrons present in an atom. So, or you can say, till electron has not involved in any bonding, till then in an atom number of proton will be equal to that of the number of electrons. So from here we can conclude that atomic number is equals to the number of protons, and it is equal to the number of electrons. Apart from all these things, there are three basic terminology we use for the atoms. These are the isotopes. So you all know that the isotopes is containing equal number of atoms as having equal number of atomic number, but they can vary with respect to their atomic number, whereas isobars are having difference in their atomic number, but their mass number will be similar. And isotones will be what? Isotones will be having both atomic number as well as mass number different, but the neutrons will be same. So as in the case of carbon and nitrogens. So why I am introducing you with all these terms, because when we come across the different biomolecules or the elements which are present in the biomolecules, we can find such types of isotopic form or isobaric forms or isotonic form inside our body also. Next thing is that the elements which are present uh, in our body basically in the larger amount consist of all these elements, which is the carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur. So these are the basic elements which all together will be going to constitute biomolecules inside our body. These elements, when they will be combined together with the help of a certain bondings, which can be of any type, like ionic bonding or covalent bonding can be there. So all these elements, when they interact with one another, it can be of similar elements or it can be of different elements, and this will form the molecules. Now these molecules are also present not only in the on the outer body in the non-living organisms, rather it is quite dominant in the living organisms too. So in this section we will be dealing how these molecules is been formed and what are the roles of these bondings inside our body, because we are starting with the biochemistry and bonding is a basic thing which we should know with respect to their biomolecules. So I am not going to explain you much about the bonding because this is basically the portion of chemistry. I will just focus on those points which will be related to your biological term, and this will be helpful to you when I will go enter particularly inside the other topics of the biochemistry like carbohydrates, fats and other topics. So moving finally to our topic, which is the types of bonding. There is no definite way we can classify bonding, but here I have classified it into two parts: intermolecular bonding and intramolecular bonding. So intermolecular bonding are basically classified into three parts: non-polar covalent, polar covalent and ionic bond. Others are the intramolecular bonding. Here hydrogen bonding can also be included in the intermolecular bonding, and other is the van der Waals forces. Apart from this, there may be other types of bondings also, like coordinate bonds, metallic bonds may be there. So in different books, in different internet sources, you might be having different types of bondings, different types of classification of bondings. In your school level you might have gone through different types of bondings, classifications of different types of bonding, but here according to my convenience I have classified it into two parts, which is the intermolecular bonding and intramolecular bonding. So come to the covalent bonding, which I will be focusing on this video. So covalent bonding is basically of two types. One is a polar covalent bond and another is a non-polar covalent bond. Here I have done a mistake. This water molecule is not the non-polar covalent bond, sorry for that. This will be included under the polar covalent bond. So what does the polar means? So when I will talk about any pillar, there will be two poles will be there, two ends are there. Similar to that of the, suppose this is a magnet. In magnet also there are two poles are there, like north pole and south pole. So when there is difference in the two polar region, in the two ends of any region, that comes under the polar covalent bond. Whereas the non-covalent bond, there is no any difference in the polarity of the two ends of any pillar. Here with respect to the biomolecules and biochemistry, the difference in the polarity means what? Difference in the electron density. So let's have a look what basically non-polar covalent bond is. First I will discuss about the non-polar covalent bond, then I will switch to the polar covalent bond and how these two are related in our body to associate biomolecules. So let's take the example of chlorine. So chlorine, you know that the atomic number of chlorine is 17 and electronic configuration you already know, this is the k element form of configuration. So you know that any compound which will undergo bonding, why they undergo bonding? So as per the Lewis, they state that any compound undergo bonding to complete their octet or duplet, or we can say when their outermost electron should be saturated. If it either it will be as in case of helium it is 2, okay, In case of hydrogen also, the outermost electron should be 2 because it is containing S form of configuration or the K form. So you know that in case of chlorine, it needs 8 electrons to saturate itself completely. So how it can be saturated? How the octet can be completed? In this case, if we will only take the outermost electron, you can see here 7 electrons are there. So it will then invite the other chlorine molecule to get saturated with the 8 electrons, and when as other chlorine molecule will also be having the 7 electrons in the outermost shell, so they will come together, and because of the coming together close together, these electron will be shared mutually to form the covalent bond. So covalent bond is what? It is the bonding formed by the mutual sharing of electrons between any two elements or atoms. So this is how we can understand the non-polar covalent. Now why it is known as non-polar? So you know that the electron around the chlorine and electron around this chlorine is almost equal, and they are having same tendency to pull electron toward itself. So there will be no difference in the density of electron around these two atoms. That's why there is no polarity will be created, and this will be considered as the non-polar covalent bond, or this in the case of Cl2 or chlorine molecule, the electron density are equally distributed around these two atoms, so it will be a non-polar covalent bond. Now what is the polar covalent bond? Here, unlike to that of the chlorine, we have one oxygen and two hydrogen molecules. So we all know that this is an example of a water molecule, and along with this, oxygen also contains two lone pair of electron around itself. So what will be the result of this? As it is containing two lone pair of electron, more electron will be attracted toward the oxygen, or it will start pulling electron toward itself. So because of this, you might have seen this sign like delta plus and delta negative. So what this sign resembles? This sign resembles, because of the pulling of electron toward the oxygen, electron density around the hydrogen will be shifted toward the oxygen. So here the density of electron toward the oxygen is more as compared to that of the hydrogen atom, and this will create the polarity with respect to the electron density. So here around the oxygen the density of electron is more. Because of this, it gets partial negative charge, and around the hydrogen there is a partial positive charge because electrons around the hydrogen got shifted toward the oxygen molecules. So this will be creating a polarity because of shifting of electron toward one side, and this will be included under the polar covalent bond. And how here the bonding is been formed? So you know that in oxygen it needs two more electron. The outermost electron is what? 6. So to complete its outermost valence electron, it needs two more electron, whereas in case of hydrogen it is having 6 electron, 1 2 3 4 5 and 6. So how this 6 electron will be converted into the 8 one? The one electron which is present in water molecule will be shared with the oxygen, and here also one electron of hydrogen will be shared with the oxygen. On the other hand, hydrogen need only one more electron to complete its duplet form, or the 2 electron will be required by the hydrogen to saturate its outermost valence shell. So because of the mutual sharing of electron between hydrogen and oxygen, this covalent bonding has been formed, and because of the shifting of electron more toward the oxygen, the polarity has been created. Now how these bondings are involved in our biomolecules? So we know that the basic biomolecules which are present in our body are the carbohydrate, fats, nucleic acids and proteins. So these are the basic biomolecules which are present inside our body. So you know that glucose, and have heard the name of glucose, phosphoglycerides and all these are being formed by the covalent sharing of electron or the mutual sharing of electrons, and they interact with the other biomolecules either with the covalent bond or by the ionic bond or by the van der Waals forces. So here carbohydrate is there. I have given you the example of carbohydrates, where you can see that there are two molecules of glucose is there, and when they have combined together to form a divalent carbohydrate. Here is a monovalent carbohydrate, and when they have combined together by the help of covalent bonding, here there is a dehydrogenation reaction has been taken place, where one water molecule has been released by the bonding of one covalent bond between two different molecules of glucose molecules, and here you can see the bond which has been formed is been given a specific name which is known as glycosidic bond. So here the bonding is covalent, but we specify such types of covalent bond with a specific name. So in the case of carbohydrate, it is known as glycosidic bond. Moving to the next biomolecules of our body, which is fatty acids. So you are well known with the two terms saturated fatty acid and unsaturated fatty acids, and you know that they are playing a very vital role in our structural body structure. In the outermost structure of the cell, plasma membrane is basically constituted of the fatty acids. It also act as signal molecules also. They are present all over our body. And how the covalent bond what is the basic function of covalent bond here? So one thing I have missed, that there may be three types of covalent bonding on the basis of number of bondings. One is the single covalent bond, double covalent bond and triple covalent bond. So here you can see that one is a saturated fatty acid. So saturated fatty acid is having single covalent bond between two carbon molecules, and this will be basically providing you a linear and compact structure. If this saturated fatty acid will be present in the plasma membrane, then plasma membrane will be packed quite close to each other, and this is quite helpful for the place where for the individual who are dwelling in the higher temperature, because it will prevent the outermost structure of the cell to melt. Whereas in the case of cold dwelling animals, it needs the separation, or if already the plasma membrane will be close compact together, then what will happen? Because of the decreasing in temperature, the plasma membrane will crystallize and the body and the cell will die. So to prevent that, there will be introduction of more non-covalent bond, covalent bond, double bond, and because of the presence of this covalent bond, a kink will be produced. So how the two plasma membrane will be interacting, or two phospholipids will be interacting when there will be kink. So when there is a linear saturated fatty acid will be there, the plasma membrane will be closely packed, but when there will be kink in these two phospholipid, then what will happen? This will prevent the close packaging of this molecule, and this will help them to survive even under the low temperature. So this is how a covalent bond, how the variation of a covalent bond from single to double covalent bond play a very vital role to maintain our biomolecules inside our body. Protein is another biomolecules which is the building block of our body. So if I say it is a building block of our body, you can now predict what the importance of protein is. Here also covalent bonding is there. It is connected, this particular covalent bond is known as peptide bonds. This will be formed between the amine group of one, or I better say this amine group of one amino acid and carboxylic acid of the other amino acid, and when there will be formation of bond between the amine and carboxylic acid, this particular bond is known as peptide bonds. So this helps to make the chain of amino acid to come together to form a primary structure of amino acid, and depending on the sequence of amino acid, secondary tertiary structure of the protein will be formed. And you know that the tertiary structure of the protein is the functional form of the protein. Here the bonding between these two amino acid and the sequence of amino acid is playing a vital role inside our body to decide the function of any protein. The protein can be of two types, globular protein and fibrous protein. So all these things I will be discussing in my class in the series of biochemistry. So all these were about the role of covalent bond in the biomolecules. Next major biomolecule inside our body is the DNA molecule or the nucleic acid. So it can be DNA or RNA, and you know that the polymerization of DNA or RNA takes place by the nucleophilic attack of one electron toward the 3' end of the ribose sugar, and these undergo condensation reaction to form a phosphodiester bond. So the bond between two monomer unit of DNA is known as phosphodiester bond, whereas the other as like that and that of the other biomolecules, as in the carbohydrate it is termed as glycosidic bond, in protein it is termed as peptide bonds. In case of nucleic acid it is termed as phosphodiester bonds. So all these were about the different types of covalent bond inside our biomolecules. Though this is not the only interaction, there may be other interaction can also be there. So these are all about the covalent bond with respect to the biomolecules. In the next class I will be dealing with the ionic bonds in the biomolecules, how these ionic bonds play, what is the role of these ionic bonds and how they function inside our body. So all these were for the today. Don't forget to subscribe and share my channel. Till then, goodbye and enjoy your lockdown.